Okay, chapter 9 is about the genetics, microbial genetics. And this is just an introduction of what we're going to do with this, in this chapter. And it's basically talking about the chromosomes and genes. The chromosomes, we will go to the sequence of each one of those, but basically the cell, if this is just a cell, not a bacteria or prokaryote, it will have a nucleus. In the nucleus we have chromosomes. Chromosomes are put together in a certain way that's composed of DNA. It's basically DNA. So chromosomes are DNA folded around a histone. We will see all these details and how the chromosomes are made of DNA, which is made of genes, and so on. We will go one by one. DNA replication. The DNA is the mother or the blueprint of all genes in the body. This is where we have everything. It's the source of the blueprint. This is what we have. And if you are going to duplicate a cell for either growing, you are replacing, or making sex cells, reproductive cells, whatever it is, you are going to just duplicate the DNA. You will make copies of your DNA, and we will call that DNA replication. And it should be exactly the same thing. I have this DNA, I make a copy which is exactly the same. I call that DNA replication. And when you replicate something, meaning you duplicate or you make a copy of it. Exactly, it should be exactly the same. Whether we're doing that for a somatic cell or for reproductive cell, and we know the difference. Somatic cell, any cell in the body, liver, skin, anything. The germ cells are sperm and ova. Whatever it is, you have to make the exact same copy. This is completely different. So what we're talking about so far is just keeping the DNA and making exact copies out of it. And we call that the replication, okay? On the other hand, if you wanted to make a protein, whether this is protein that will be used in the structure of the cell, or you're making a hormone, or whatever the protein is, you're making a protein, or even an RNA, whatever you're doing, you are going to do a different thing, which is you're going to make a copy of part of the DNA. And we call that transcription. So replication is different than transcription. Are we following? Replication, you make a whole copy of a whole DNA exactly the same, okay? And this is for the purpose of keeping the same DNA, you're duplicating your cells, you're making the germ cells, whatever it is. You're making the exact copy of the whole DNA, and we call that replication. If you wanted to make a protein, on the other hand, you make a copy of part of the DNA, and this copy of part of the DNA, we call that process transcription. So transcription is to make what from what? Which RNA? Messenger RNA. You make messenger RNA out of DNA. Out of the whole DNA? No, out of part of it. Not the whole thing. Part of the DNA. And then this messenger RNA is going to leave the nucleus, if this was a cell, and it's going to go to the cytoplasm where the ribosomes are located. And you're going to do the next process, which is the translation. And translation is to translate the transcript into a protein. So DNA, can you tell me what you call it? You make DNA from DNA. What is it called? Replication. You make messenger RNA from DNA. Transcription. You make protein from the transcript that you have, which is the messenger RNA. We call that translation. Mutation is a change in the code. We're not supposed to have any changes. This DNA, whether you are doing a replication, you're making DNA from DNA, or you're making a transcript, which is called a transcription, you're making messenger RNA from DNA, we're supposed to have it exactly the same. If it is DNA from DNA. If it is messenger RNA, it should be the exact correspondent to the original copy that you have. If something happened to that code and changed for some reason, we will call that mutation. And because of that mutation, of course, the transcript will be translated wrong, right? Here is my DNA. I have this 1, 2, 3, 4, 5. I have code for 5 amino acids, for example. You make a messenger RNA for those 5. You translate for those 5. If something changed, whatever the level is, if something changed in that code, whether you're making a wrong copy, which is a transcript, or the transcript itself become wrong for some reason, you're going to translate it into wrong protein or no protein, right? Doesn't have to be the wrong protein. It can be no protein. In all cases, most of the time, this is something that's usually harmful. Occasionally, it's not harmful. We will see. But most of the time, it is. This is if something happened to any code. DNA code. Recombination, simply re means again, combination means to combine. So recombination means to combine DNA from one cell to the other. This is recombination. You get it? I get part of this DNA and I recombine it to another DNA from another cell. I call that recombination. You can take DNA from bacteria to another bacteria. You can take it from virus to bacteria, from bacteria to virus. These are different ways if you're doing that in the lab. But it can happen automatically. You don't have to do it, right? Maybe this bacteria will go to another bacteria and it will do automatic recombination. You didn't do it, right? So recombination is whatever the way it's done, it's you're combining part of DNA from one cell, recombining it to a DNA from another cell. Whether this is happening automatically or you are doing that intentionally, whatever the reason or whatever the technique is, in all cases, it is recombination. Okay? Genetics in general is the study itself. You're just studying. So we have different terms here that we need to understand and know as we go. So genetics is the study itself. You're studying everything about the hereditary, about the genes, everything. How it is transmitted, how is it translated, how is it expressed in the form of traits. What traits mean?
Specific characteristics.
To express what?
Like a specific characteristic.
Yes. Like the yellow hair is a trait. The dark hair is a trait. Whether you're short or tall, this is a trait, right? The color of the eye is a trait, and so on. So how is it going to be expressed? The function, the structure of the genes, any changes that can happen. So all this study is called genetics in general, okay? And the level of the structure of the genome is, if you look at the first one in the left, this is the whole organism. Introbius, this is one of the helminthes, they're studying it. So you take one cell, this is the cellular level, you go into the nucleus, you can take one chromosome, and if you unfold that chromosome and look inside, you will see this molecular level. So you go from organism to cell to chromosome to molecular level. Look at this here. This is the second part. You take the chromosome, you look inside of it, you will see this sequence, and this is something that we have to understand. This part is called the gene. It's part of the DNA, and each gene will consist of codons. We will discuss all the details. This is just introduction in the beginning. So here is a chromosome. If you look inside of this, you will see gene after gene after gene after gene after gene. Okay? If you look inside of that gene, one gene, like this, you will see codon, codon, codon. What's a codon? It's H3 nucleotides. And this is one of the nucleotides. So let's do it like this. Here is a whole cell, right? This is the nucleus, okay? What's the nucleus contain? It contains chromosomes. Is that okay? I will take that chromosome and unfold it, okay? Just straighten it and look at it. What I'm going to see within this chromosome, DNA. Let me look inside the DNA. I will see patches. Each patch is a gene. Is that okay so far? Now I will look inside the gene. Each triplet. The gene contains a lot of triplets. Each triplet is called a codon. What's the codon? It's a triplet. Triplet of what? Of nucleotides. What are the nucleotides? We will discuss it. Okay? But you go from chromosome to DNA to gene to codon to nucleotide. Is that okay so far from the big level to the small level? Because we will have a lot of details, so just follow as we go. So genetics is the studying of hereditary or the genes. What's the genome? Genome is the total of the genetic material, which is the DNA inside a cell. Everything that you have all together, we call it a genome. Okay, usually in the form of chromosomes. This is what we're going to focus on, but for your information, we should also count the genes that's located somewhere else. Do I have genes somewhere else beside the chromosomes? Yes, a little bit. You can have some in the mitochondria, chloroplast, or plasmid. Each one of those, it's not considered a chromosome, but does it have genes? Yes. So where do we have the vast majority of the genes, which make the genome? In the chromosome. Do we have genes somewhere else, not in the chromosome, which is 90-something%? Yes, we can have it in the mitochondria, chloroplast, and plasmid. Okay? So the genome of the cell is the DNA. This is if we're talking about cells. Which cells? Any cell. Any cell. You have DNA. This is genetic material, okay? Or the genome. Viruses are different. Do we consider virus, we consider it cells? Is a virus cell? No, it's not a cell. The virus is not a cell. But it does contain genome. And it can be DNA and RNA. And we did the viruses already. Remember that chapter? Some of them will be DNA and some of them will be RNA. But can we say genome, RNA in humans? No, genome in humans or any cell, not necessarily humans, is DNA only. So the only exception is viruses. So the chromosome is a packed DNA molecule, a big molecule that's packed. Why is it packed? Because you need to put it inside of the nucleus. You know how actually the chromosome, if you unfold it, it will be as tall as me. Think about it this way. It's as tall as me. Okay, about 6 feet. This is so you have to pack it all together and tighten it to put it in that very tiny space, which is the nucleus. Okay? In that matter, eukaryotes and bacteria are different. Prokaryotes and eukaryotes are different. How is it different? Do you remember in prokaryotes, do we have nucleus? We don't have nucleus in the prokaryotes. We do have chromosomes, but it's not inside the nuclear membrane, right? Do you remember what's the most important thing about the prokaryotes? And when do you call it prokaryotes? You don't have nuclear membrane, so it's not a nucleus, and you don't have membranous organisms. Wasn't that the difference? The main difference. So inside the eukaryotes, I have the chromosomes inside the nucleus. While inside the bacteria, do you have nucleus in the bacteria? No, so it will be inside the cytoplasm. It will be inside the cytoplasm. This is one. Number two, in eukaryotes, we have multiple chromosomes, a lot of chromosomes. In bacteria, you have one. The other thing is, how does it look like? In eukaryotes, it's linear. In bacteria, it is circular. Are we following? Linear versus circular. And this is very important comparison between the chromosomes in eukaryotes and prokaryotes. This is very important, okay? In eukaryotes, the gene will code for one protein only. And in prokaryotes, it can be different than that. It can be for more than one. Okay, are we following so far? These are the differences. There is a difference between genotype and phenotype. When you say genotype, what do you mean? And when you say phenotype, what do you mean? When you say genotype, you're talking about the genes that is transmitted from parents to the offspring, right? This is the genome. So this is the genotype. So if you're telling me what's my genotype, I will tell you details about your genetics, okay? About your chromosomes. If you're talking about phenotype, phenotype is the trait, okay? So if you tell me like something like this, my hair is black, okay? My hair is black. Can you tell me what's the genotype and phenotype? I will tell you yes. The genotype is the genes that are responsible for hair to be black, for your hair to be black. The black hair itself is the phenotype. Did you get the difference? Genotype versus phenotype. So this is the difference between those two. And the genes in general, the vast majority of the genes are going to code for proteins. If that's the case, we call those structural genes. But these are not the only genes. These are the ones that we're going to focus on. Okay? But these are not the only genes. And what's the gene? Are you following?
The genetic.
What's the gene?
The genetic.
It's responsible for a trait.
It's responsible, not necessarily a trait. The DNA is responsible for a trait. And the DNA is part of the DNA. But what's gene specifically means?
A specific trait.
A specific triplets that code for one protein. A specific triplets. Do you remember this picture that I showed you? Finding the chromosome, and then you unfold the DNA, and this part of the DNA is called a gene. And this gene consists of triplets. And each one of the triplets, the triplet is called codon, right? And each codon is 3 nucleotides. Did you remember this picture? Yes.
So the gene is like a strand of the DNA?
Not a whole strand. Part of.
Okay.
Part of. It's exactly like this. This is the whole chromosome, okay?
Mm-hmm.
If you unfold it and look at it, you will see it's a DNA. Okay, so far? This part from here to here, this is a gene,
And it's a group of 3, you said?
which is a group of 3. So if I put it like this, it's 1, 2, 3, 1, 2, 3, 1, 2, 3. Are we following? So this from here to here is a gene. From here to here is another gene. So this is 1, 2. From here to here, 3. Did you get it? So from here to here is gene. Here it is. Why is it from here to here? From here to here to be responsible for making a protein or a trait. Okay? So let me look more under the microscope. Under one gene. And the DNA contains a lot of genes. Are we following?
Mm-hmm.
So let me look more under the microscope. You will see triplets. Each one of those is called, each one of the triplets, a codon. And what is each one of those? Nucleotide, not the basis yet. Nucleotide. Is it confusing?
Yes.
Chromosome is DNA folded and tightly coiled around histone. Okay for this? This is a chromosome. Let me look inside the chromosome. I don't care about the histone now. Let me unfold it. It's folded around the histone, right? Unfold it. And let me look at it. What is this? It's DNA. Okay? Chromosome is DNA tightly coiled around the histone. Is that okay? DNA around histone. I don't care about the histone. Show me the DNA. What is that DNA? It's a double huge strand, 2 strands, and that consists of Gene after gene after gene after gene after gene. What's a gene? It's a segment of the DNA that will code or that will be translated into one protein or a trait. Is that okay? So let me look inside of that gene from here to here. It's triplets. Each one of these triplets mean 3. Each triplet is called a codon that will code for amino acids. We will go to the details. And what's a triplet? 3 what? Nucleotide. We will go to the structure of the nucleotide later, but I want to make sure that you got the big thing. Chromosome is DNA around histone. Is that clear? Unfolded from the histone. Show me what you have. DNA. Inside the DNA, I have gene after gene after gene after gene. What's a gene? It's a group of triplets. What are these triplets called? Codon. What's each codon consists of? 3 what? Nucleotides. We will discuss the structure of the nucleotide, but so far, are you following with me? From large to small. So, genes, like this segment right here, it can code for protein. This is the structural gene, and this is what we're going to focus on. Okay? But it can also code for RNA for another reason. Okay? And it can control the gene expression. We call that regulatory gene. So this gene doesn't have to be translated into a protein. It can make RNA. It can be controlling the expression of the genes, which we call it regulatory genes. So we have 3 types of genes. Even though we're going to focus more on the ones that make the protein, which is called the structural genes, but know that there are another 2 types of genes. And what's a gene? It's a segment inside the DNA. Okay? So here again, it's a genotype versus phenotype. Genotype is the genetic makeup. Phenotype is a translation of that in the form of trait. Okay? The size of the genome, and what's the genome again?
It's all genetic material.
Yes. The sum of all these genetic material. Okay? So the virus, for example, contains 4 or 5 genes. Very tiny, very small. 4 or 5 genes, that's nothing. E. coli, which is a very simple bacteria, it will contain about more than 4,000. So you don't have to remember the numbers, but know at least that virus just contain few. E. coli, it's just a bacteria and contain 4,000. How about humans? 31 genes. We have 46 chromosomes, 31 genes in one cell only. And if you unfold it, it will be as tall as me, 6 feet. Okay? Let me say something here. To be clear, every single enzyme is important. To the extent of when you see these enzymes, write it down in a piece of paper or something and memorize it. Extremely important. Any single enzyme. When I mention enzyme, extremely important. Okay? We have to know all the enzymes. So the DNA is super coiled. It's coiled around the histone. And why did you coil it and make it that tight? To fit into the nucleus, which is very tiny. Imagine that you have to fit 6 feet long inside this nucleus, which you can't even see, right? So we have to tightly coil it. How is it tightly coiled like this? What keep it in that? Why it doesn't like unfold by itself? In prokaryotes, there's something called DNA gyrase, and this is the one that's going to keep it like this. In eukaryotes, on the other hand, it is DNA coiled around histone, and what's keeping it like that is the very complex structure. It goes level after level after level, so you don't need something specific to keep it like this. It's the way it was coiled that will keep it as it is. And it's DNA around histone. Remember that the chromosome is DNA around histone? Chromosome is DNA around histone. Tightly coiled. So you wrap it, you super wrap it, 3 different steps in order to end up having this that will not unfold unless you want to unfold it. So again, if you look at this picture right there, here is chromosome. Okay, chromosome is a tightly coiled. This is chromatin, which is loosely coiled. If I look at it, it contains fibers. These are called the nucleosomes. Nucleosome is DNA around histone, and this is DNA. Or let's take it the other way around. Here is the DNA. If you unfold it, okay, it's coiled around histone, right? Here is one more name. Chromosome is DNA around histone. Is that clear? Chromosome is DNA around histone, tightly coiled. Around histones, a lot of histones, right? But each coil around one histone, I call that nucleosome. Are we following? Okay, so how many nucleosomes? A lot. Depend on the number of histones. So if you unwrap it together, or if you wrap it together like this, it will be a chromatin fiber. A lot of chromatin fibers will make the chromatin material, and if it is tightly coiled, you call it chromosomes. Now we went all the way to this level, right? To the nucleotide. Are we following? Chromosome, DNA around histone, contain genes, triplets. Each of those triplets is a nucleotide. Now, okay? Now we came to the point of nucleotides. Whether you're talking about DNA or RNA. By the way, this is DNA, right? How about the RNA? It's just one of those. The RNA is one, and not even the whole one, part of it. Okay? So whether we're talking about DNA or RNA, it consists of nucleotides. Each three will make a codon. What's a nucleotide? It depends. When you're talking about DNA or RNA, we're talking about DNA. Let me say this. Where did you get the name from? DNA. Deoxy. Okay, deoxy. R is ribose. It's a sugar, 5-carbon sugar. DNA, deoxyribonucleic acid. This is what DNA means. Deoxyribonucleic acid. Deoxyribonucleic acid. What's deoxyribo? Deoxyribo is ribose, and you removed oxygen. Does it make sense? What's deoxy mean? Without oxygen, right? So here is ribose, which is a sugar, 5-carbon sugar. You take oxygen out of it, I call that deoxyribose. So what kind of sugar? Ribose or deoxyribose? Deoxyribose. So obviously RNA means ribose. So we're talking about the structure here. Sugar, it is the general structure. We have sugar, phosphate, and base. Sugar, phosphate, and base. What kind of sugar and what kind of base? This is different from DNA to RNA. We're talking about DNA now, so the sugar is deoxyribose. What's the sugar in RNA? Ribose, yes. So is the sugar different from DNA to RNA? Yes. In the DNA, deoxyribose. In RNA, ribose. And this is where the name came from. What do you mean by RNA? Ribose nucleic acid. What's ribose? So the first part is the sugar part. Phosphate is not different. It's just a phosphate, right? Nitrogenous bases, we have 4 different nitrogenous bases. What do you mean by nitrogenous base? It's a base that contains nitrogen. And we have 4 of those in the DNA. Adenine, guanine, thymine, and cytosine. Okay? From the chemical point of view, adenine and guanine are called purine. Thymine and cytosine are called pyrimidines. This is in DNA. In RNA, it's uracil. This is from the chemical point of view. We don't have to know the details. What do you mean by purines and what do you mean by pyrimidines? It's just a chemical category. They just belong to. So adenine and guanine are called purines. Thymine, uracil, cytosine are called pyrimidines. With pyrimidines. Okay, so far? So nucleotide and another nucleotide, this will make, each 3 will make a codon, and we will make the gene and chromosome. But nucleotides are bound together by covalent bond. to form sugar phosphate backbone. What do you mean by that? Sugar phosphate backbone is the sugar, which is deoxyribose in this case. You added phosphate to it. Sugar phosphate backbone. This is the backbone. This is the back of or the backbone of the DNA. Okay. Which is purine? Which is pyrimidine? You need to know that. Ag is purine. TUC is pyrimidines. These nitrogenous bases, the nitrogenous bases, are also bounded by covalent bonds. Together by covalent bonds. Okay, so you make it the same strap like this to this to this. Can you see this? This to this to this, covalent bonds in between those. Okay? This is one strand. Same from the other strand. And how the two strands stay together like this? Hydrogen bond. Okay? Hydrogen bond. For A will fit into the T and C fit into the G. And the bond in between is hydrogen bond. What's holding the nitrogenous bases together? It's a covalent. Do we remember the covalent versus hydrogen bond? So yes, so the nitrogenous bases are bound together by covalent bonds in the same strand. But if you are talking about the two strands, how the two strands stay together, hydrogen bonds. If anything is not clear, let me know. I know this is kind of complicated. Genetics is usually not easy, but we need to understand it. This part is extremely important. What fit with what? Here is I have one strand, here is the other strand. What fit with what? Nitrogenous bases. A, T, C, G. At C, G. At C, G. A fits with the T by hydrogen bond. C fits with the G by hydrogen bond. Is this part clear? You have to remember this. At Cg. At Cg. This is how they fit together. How they fit together in the two strands. Okay? So look at this here. Here is the A from one strand fit with the T. C with the G. G with the C. It's the same thing. T with A, right? Whatever the sequence is, they stay together by hydrogen bond. But this backbone right here, which is the phosphate sugar, what's holding these together? Is it a hydrogen bond? What is this? Covalent bond. You know why? Because you don't want to cut this from each other. Remember that the covalent bond is the strongest bond? You don't want to cut this. But can you separate the two strands? Yes, you always do that. Which is a hydrogen bond, it's a weak bond. The way the two strands stay together is called the anti-parallel arrangement. There is one end of the strand that's called the 3 end, and the other one is called the 5 end. But they fit together. Look at this to understand what I'm talking about. Here is one strand, and here is one strand. My fingers here is the 3 end, and this is the 5 end. Is that okay? If I put together like this, I call that parallel. But look at this, it will fit like this. So anti-parallel. 3 end, 3 end, 5 end, 5 end. If they are going to fit, which is not going to happen. If they fall together like this, I call that parallel, which doesn't happen. What's actually happened is like this. I call it anti-parallel. So 3, 5, 5, 3. Is that clear? It's anti-parallel. This is how it stays together. The code should be maintained during reproduction. That base pairing that we're talking about, that goes like this, and how they fit together in a hydrogen bond, should stay together in reproduction. So if you're going to make a gamete or something, it should stay together. The two strands that are going to be replicated later on are called the template, but we will talk about it a little bit later. And it will provide varieties depending on the sequence of bases that will give us different varieties. Can you tell me this? Can you answer this question? Is it A or C? C. Yes. Okay, answer this. Yes. So this is something I left it like this, so I see if you're going to get it or not. It's usually say, oh, T, T, A, A. No. There is no T in RNA. It's U. Okay? This is how the mistake can come. Remember this. If I'm giving you a sequence, you need to remember, are you talking about replication or transcription? Is that clear? If you're talking about replication, it will be TT. Right? If you're talking about transcription, you're making messenger RNA. There is nothing called thymine in the messenger RNA. It's called uracil. Okay? All right.
So if they gave us, never mind, sorry.
Okay. All right. So now the replication itself, how is it going to happen? Remember that the two strands are winded together like this. It's not like this. Look at this. It's not like this. It's like this. It's winded like this, twisted. Okay? So you're going to unwind it, unzip it, and then you are going to replicate the two strands at the same time. I will make a copy of this strand and this strand. Am I going to make a copy of this and after I'm done I'm going to make a copy of the other one? No. You're making copies of both of them at the same time. Is that clear? Like this, look. Here are the two strands. This, copy post at the same time. Okay? And you're going to make two complementary strands. So you call this process semi-conservative. What do you mean semi-conservative? I mean, these are the two strands. I'm going to separate them, right? This is one template, and this is one template, right? And I'm going to make a complementary strand to this one. So how much does it have from the original? It's become two now, right? I'm going to make one from this and one from this. This will be a DNA, and this will be a DNA, right? This had one of the two strands original, and this have one of the strands original. So we have one original and one copy. I call that semi-conservative. Semi-conservative. Okay? The original one, both of them are the templates. But what's going to happen is I will have a copy and copy. So look at this. These are the original strands, the templates, right? Look at the red. The red is the copy. So you end up having two DNA. But look at this. This is semi-conservative. It has original, which is blue, and red, which is a copy. I call that semi or semi-conservative. Where exactly are you going to start the replication? There is a spot that's called the origin of replication. Origin of replication. This is exactly where the replication is initiated. Where do you start? Origin of replication. Okay? If you are talking about the transcription, this is if you're talking about replication. What if you're talking about transcription? Transcription will occur when something called RNA polymerase attach to the region that's called promoter. So this is not supposed to be here. I just put it for comparison. If you're talking about DNA replication, do you understand the difference between replication and transcription? If you're talking about replication, where do you start? Origin of replication. Done. What if I'm talking about the transcription? Where do you start? Promoter. I start at the promoter. Where the DNA attached, RNA polymerase attached to the promoter. Okay? You make something, if you're starting this process, you make something called replication fork. Fork is like this. Okay? It was like this, but when you separate the two strands, not the whole two strands, it goes like this. Look at this. I'm going to unwind. I didn't separate yet. And I'm going to separate exactly like this. Look. Right? So are you making a fork now? This is a fork. It's not like this. No, it goes like this. So as you are separating, you're making a fork. What's fork is like this. This. And we call that the replication fork, in which you're going to do the replication. So the first thing that you're going to do, you're going to unwound the chromosome, and the enzyme that's going to help you with that is called helicase. Are the enzymes important? All of them. Every single enzyme is important. So helicase, if you forgot it, understand what does it mean. Okay? Helicase. Ase means an enzyme that works on something. Helic means the helical, which is this. This is called helical. So helicase is an enzyme that will break down the helix, or it's going to unwound it. Okay? You will have two strands. These are called the leading strands. Leading strand. So what's a leading strand? The template. The enzyme that's going to help you with this process, then when you start to make the replication itself, is called DNA polymerase. So what's the difference between helicase and polymerase? Helicase, if you break down the helix, right? You're breaking down the helix. Polymerase, polymerase is coming from polymer. Polymer means you're making a copy. Polymer, okay? It's these nitrogenous bases, you're making copy. We call it polymerase. So polymerase is the one that's going to make the copy, which is the one that's going to do the actual replication. Okay? So polymerase is the one that's polymer. What do you mean polymer? I mean a lot of nucleotides. A group of nucleotides, polymerase. So it's going to add the nucleotides in a 5'3" direction toward the replication fork. I have a video for that. I will show you so you imagine more what I'm talking about. It will do it from the 5'3" direction, and this will be a continuous strand. On the other hand, you're making the other leading strand will make another copy. DNA polymerase will make it, but in the other way around. The lagging strand, which is the other strand, will proceed backward, away from the replication fork. Okay? So in that case, you're making fragments. And these fragments are called Okazaki fragments, and you need to remember those. Okay, I know you didn't imagine exactly what I'm talking about, I'll show you. And then you will join these fragments together by something called ligase. If you forgot it, ligase is coming from ligation. You know what ligation means? When you ligate something means you put them together, you attach them together. This is what ligase means. So let's see the video to have a better understanding. So remember again that you are going to make copies it's semi-conservative. Here is the template and here's the other template, right? I'm going to make a copy. What is the name of that copy? It depends. The one that come from the 3-5 direction is called the leading strand. The other one is called the lagging strand. The leading strand is continuous. The lagging strand is not continuous. Okay? and then you're going to put them together by the enzyme ligase that's going to attach these fragments together. Enzymes that we use, we started with helicase that's going to unwind and unzip, DNA polymerase that's going to add the nucleotides, but it's two different ways. Leading strand is continuous, lagging strand is fragments that are called segments or fragments that are called Okazaki fragments, and then ligase is going to ligate them together, meaning hold them together. So here's helicase, DNA polymerase 3, and ligase. Can you answer this question? Yes. Okay. The next part will be the transcription and translation. We talked about replication already, right? Now transcription. What's transcription? You make what from what? Yes. Which RNA? Messenger RNA. Yes. So if you look at this here, here's the DNA. Look at this. I think it will make the picture more clear. The chromosome is DNA around histone, right? These are the DNA. Look at this. This is gene, right? And then when you do the transcription, this is a DNA template. You're making messenger RNA, and you call this process transcription, right? And the transcription, does the messenger RNA have T?
No.
It's U, right? So anytime you see A, you're supposed to put T. No, I will put U. This is the transcription. And then this transcription with the codon, what's the codon? The triplets. Triplets of what? Nucleotides. So these are triplets, and which is called the codon. It will go to the ribosomes, and you're going to translate. Each codon will make one amino acid, which is the triplets. Okay, each one of these triplets, 3 bases. This is called a codon that will be translated into one amino acid. And then look at these amino acids. So let's say codon here, this make tryptophan, one of the amino acids. And the next one make phenylalanine. And this make glycine and serine. So you have amino acids, right? These amino acids, you're going to hold them together to make a protein. What's in the protein is a group, a big chain of amino acids. Do you remember this? So you're producing them in the form of amino acids, and then you hold them together. So remember that the triplets on the nucleotides, the triplet is called codon, right? And each one of the codon will be translated into one amino acid. What do you call a group of codons together? Like this patch here of the DNA that have these codons. Gene. Gene is a group of codons. The code or the codon represent amino acid. The gene represent protein. Is that clear? So what's the difference between DNA and RNA? We talked about those. Okay, let me ask you, which one is double strand and which one is single strand? DNA is double and RNA is single strand. Okay, this is one. Number two, what kind of bases do you have in the DNA? The 4 bases. A, T, C, G. And what kind of bases do you have in RNA? AUCG, right? So you don't have thymine, you have uracil instead, right? How many RNA do you have? We talked about one of them, but how many do you have altogether? 3. I didn't mention that, but I'm telling you. You have 3. Messenger RNA is the one that's formed by transcription, right? You have another 2 coming. Ribosomal RNA and transfer RNA. You have 3 different types. And last question, what kind of sugar do you have in DNA versus RNA? Deoxyribose versus ribose. Okay, great. So this is a comparison right here. Here is a DNA, ATCG, which is what you guys mentioned, right? ATCG versus AUCG. This is deoxyribose, this is ribose. See, here is a messenger RNA that have triplets that are called codons. And so this is a messenger RNA. We talked about it. The second type of RNA is called a transfer RNA. What's a transfer RNA? Another RNA that acts as a translator for the messenger RNA, and it is going to grab amino acids for you. It's doing two things: translator and carrier. Transfer RNA. It's a translator and carrier. This is the one that's going to transfer. Okay? How does the transfer RNA, since it's going to translate and grab you the amino acid that you need, how did it know which amino acid you want? They have an anticodon that fits into the code. Are we following? So we have this code, AUG. What does it mean? Which amino acid? The transfer RNA that fits into this triplet, he will translate, and when he fits by anticodon, he's going to translate first and then bring the correspondent amino acid depending on the code. So which RNA have the code and which one, codon, and which RNA have the anticodon? Which one have codon?
mRNA.
Messenger RNA, right? Right. And which one have the anticodon? tRNA. And what else is it going to do besides translating codon versus anticodon? Carrying the amino acid that you need. So this piece right here is called the anticodon, right? And this is what's going to fit with the messenger RNA. What's the third one? It's called the ribosomal RNA, which is in the ribosomes. And what's the ribosomes? It's ribosomal RNA with protein. What is the ribosome? The ribosome is ribosomal RNA packed with protein. So there is a difference between ribosome and ribosomal RNA? Yes. Ribosome is ribosomal RNA packed with protein. Is there a difference between chromosome and DNA? Yes, chromosome is DNA around protein. Is that clear or not? Did you get the difference? You cannot say that chromosome is the same as DNA, because chromosome is DNA around protein, which is called histone. The same way, you cannot say ribosome is the same as ribosomal RNA. It is not. It's ribosomal RNA around the protein. Both of them. Clear? Okay. So what is it the ribosomal RNA is going to do? Why do we need it? You have messenger RNA that have the codon and the transfer RNA that have the anticodon to fit, and it will get you the messenger RNA. Why do you need a third one? The third one is going to hold those together, like housing this whole process. You get the idea? I have a codon here, and the anticodon will come. Where? What is going to housing those? Who is going to keep those together? It's a ribosomal RNA, which is in the ribosome, which consists of two components. One is big and the other one is small. Okay? So how are you going to translate? Which is called the gene expression. How are you going to express? Gene expression. You're going to express, meaning translate. Clear? Gene expression. What's the gene expression? You're going to express the gene. What do you mean? To translate it. Transcription. Translation. How are you going to start this process? You're going to start with the initiation first. RNA polymerase is going to hold to a promoter. I mentioned that before, right? So what's your very first step if you wanted to translate? RNA polymerase. This is an enzyme, right? It's an enzyme, so it's important. RNA polymerase. RNA polymerase is going to bind to a promoter. Okay? Then you're going to make elongation. RNA polymerase is also going to add nucleotides complementary to the template until you come to the point which is called termination. When you see a code that means stop, this is where you end. Like this. I'm going through this, okay? Go, 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 until what? Until I see a sign that says stop. What is that sign? You will see it. Now, what was the codon again? It's code for what? What's the codon codes for? Codon not for protein. Gene is for protein. Codon is for?
Trade.
Or? The code. This. Amino acid. I know it's kind of confusing, but look at this again. I will repeat again, don't worry. The chromosome is DNA around histone, is that clear? I'm talking about the DNA now, I don't care about the histone. DNA, it's a double strand, right? Piece after piece after piece after piece. Each piece is called gene. The gene corresponds to a protein. It carries instructions for one protein, clear. Within the gene you have codons. Each codon will mean amino acid. Isn't the protein is a chain of amino acid? Isn't it? Okay, so the gene corresponds to protein. The codon corresponds to amino acid. Is that clear? Okay. So the codon will mean, each codon will mean amino acid. Here is something that they found to be interesting. You do not need to know all these codes. There are 4 codes that you have to remember, but you will see a lot of codes. You don't have to know the codes exactly. Just look at it to understand what we're talking about. They found this interesting piece of information that they found that some amino acids will be coded by more than one code. It's not just one code. Okay? So UAG and CUG, both of them will make the same amino acid. And we call that, the code in this case is redundant. Redundant. It does not necessarily one code for one amino acid. What redundant mean? More than one codon for the same amino acid. When you see it, you see codon number 1 or number 2. In both cases, I will bring the same amino acid. Is that clear? This is where we call it redundant. It's redundant. It's not one for one. It's sometimes more than one for one. So look at this. Obviously this is too much. Okay? But look at this here. Look at this amino acid. Histidine is amino acid. How many codes do we have for the same amino acid? You see 2? Look at this. For histidine. This and this. So when I see this, I will get you histidine. What if I see this? I will also get you histidine. So both of them code for the same thing? Yes, I call that redundance. Is that clear what redundant means? More than one code for the same amino acid. Here are the codes that I want you to know. 1, 2, 3. AUG, I remember it's WAG, WAG, WAGA. You can call it whatever you want, but you have to remember it. AUG is to start. AUG. Translation. AUG. Translation. Start. Okay? WAG. WAG. WAGA. 1, 2, 3. Stop. You have to remember this. Other than that, just understand the idea that it can be more than one code for each amino acid. Okay, this will be too much to memorize all these codes. Just the 4 codes: 1 for start and 3 for finish. So now we're going to interpret these codes into amino acid. Transfer RNA is going to come, fit its anticodon to the code on the messenger RNA. It's going to bring the amino acid, and then you're going to hold these amino acids together to make the protein. So you will start with when you see the AUG, you're going to start. and then you go from 1 to 1 to 1. Each 3 will be each 3 will mean one amino acid, which is a code. Transfer RNA is going to bring it. And then each triplet, which is a codon, mean amino acid, right? So I see the first 3. Look at this here. This is a messenger RNA, right? What is this? It's going to fit in here. The anticodon. So what is this? No, this is a transfer RNA. What have the anticodon? Transfer RNA, right? So here I'm coming. Let me fit into this codon. I have the anticodon, right? Fit in. Yes, I can fit in. So anticodon, my anticodon versus the codon. So I'm the one responsible for tryptophan. I'm going to bring it. So look at this. Here, fit, amino acid. The next one will come and fit, another amino acid. The next one will come fit, and another amino acid. So I have amino acid, amino acid, amino acid. But these are separate, right? So I need to hold them together to make a protein, right? The bond between the amino acids is called a peptide bond. Okay? And then when the transfer RNA bring the amino acid, it's going to be released and go back to the cytoplasm. How long are you going to go? Until I see the stop, which is wa, wag, waga. Wa, wag, waga. Okay? Any one of those 3. When I see it, I have to stop. That's it. I'm done. So you translated now into 200 amino acids. And then you see the stop sign, that's it. Then you're going to hold the amino acids together to make the protein. Here are the termination codes again. And these termination codes does not correspond to transfer RNA. Are we clear? The wa, wag, waga, these termination codes, the stop codes, is not going to be translated. So if there is a transfer RNA for any one of those 3, no. When you see it, you stop. You don't get a tRNA, and you do not get an amino acid anymore. You're done. Once you see it, just read. Oh, I see this sign. So if there is an anticodon for this, for tRNA, no, there is not for those 3. Okay? So let's just watch this. Video. I hope it will make it more clear. Okay, here is one thing that's important to remember, that so far we're talking about transcription that happened, then you have messenger RNA, right? And then the messenger RNA leave the nucleus and go to the ribosome where the translation occurred, right? This is not the case for the bacteria, for the prokaryotes. Why? Because you don't have nucleus in the first place. So you translate at the same time. Translation and transcription happen simultaneously. Do you get what I mean? You're making the messenger RNA and I'm translating at the same time. This is only for prokaryotes, which is bacteria. Which one of those is right? The translator and carry amino acids. Right? Okay. Codon, anticodon. Can you give me, this is a codon for threonine. What's the anticodon? UGC. Yes. In prokaryotes, bacteria, for example, they have the way the process is regulated is through something called operon. Operon is coming like from operating something. It's a group of genes that it's going to regulate only in the bacteria or prokaryotes. So the operon can be inducible or repressible. Okay, inducible means it's not working, it's off. but you can induce it. It's off, this is normal, it's off. But you can induce it. So if you need it, induce it. Like the lactose operon, for example. If you want it lactose, you have to switch it on. It's off already. The other way around, or the other type of operon is repressible. It's working all the time, which is something obviously you need all the time. But you can turn it off, which is repress it. You can repress. It's working, but you can stop it. Inducible, it's off, but you can turn it on. So can you answer this? Yes. Okay. Okay, now mutations. Mutation, again, can occur in any level, whether during replication or during transcription. You can do a mistake, right? Mistakes can happen. Which is the basis. The sequence can change, mutate, the nitrogenous base, like this. This is the code, right? If you change something here, this will lead to, which is the genotype, right? It will lead to a different phenotype. So here we're going to call the natural non-mutated type, we'll call it the wild strain or the wild type. If it is mutated, we call it the mutant strain. Okay? Wild is the normal that does not have any issues. Mutant is the one that's mutated for some reason. Okay? And obviously the phenotype that will be produced because of that will be different according to the type of mutation. Mutation can happen spontaneously, but it can be also induced. What do you mean? Spontaneously, it just happens. You don't know why. It's random. Something happened during replication. You did a mistake, right? And it happened automatically by itself. Nothing induced it. The other type is called induced. There is something that's called mutagen. Mutagen. What's muta means? Mutation. What's gen? Generation. So this is something that's going to generate mutation. It's going to induce mutation. What is that something? It can be a physical, it can be a chemical compound. Sometimes it can be even a virus. So, and that's why, for example, we always tell you, if you are in an airport and you're traveling a lot, try to avoid the X-ray, right? If you travel every now and then, that's fine. But if you travel on a regular basis, like some people every 2 weeks or something, they travel all the time, try to avoid the X-ray. Why try to avoid the X-ray in the airport? Because the X-ray is mutagen. It can induce mutation. You're exposed to that so many times, it can lead to change in that code, and it will lead to a mutation. This is just something to take a look at it. It would be too much to ask you about this. Just look at it. Mutagens. Chemical mutagens, radiation mutagens. Something like nitrous oxide, for example. Something like analogs. Something like the X-ray for radiation, ultraviolet ray. Like they always say, you're not supposed to stay in the sun or exposed to ultraviolet radiation a lot of times. You can get skin cancer, right? Why? Because the ultraviolet, in this case, is going to cause a mutation, right? Ultraviolet specifically is going to cause cross-link between the pyrimides. This is what's going to. This is too much details. Just look at it and understand what we're talking about. Whatever the change is, you are going to link the pyrimidines together, or you're going to remove something or add something. Whatever it is, it's going to lead to mutation anyway. And the mutation can be of different types, and this is very important here, the slide to remember, and we need to understand it first. Point mutation is you're mutating at a point. And when we say point, we mean the base, one of the bases, one or more. Okay? What are you going to do with the bases? Look at this. What if I did this? Deletion, right? I can delete one of the bases. Or put it back. I can add like this. Right? You can delete, you can add one or more. So we call that point mutation. Point means base. You're doing something with the base, whatever it is, adding, deleting, whatever it is. Missense means when you change something, like I'm going to look at this here. This triplet here means methionine, okay? If I remove this and put another one here, switch, it will be translated into another amino acid, and we call that missense. Missense. So you're missing, just to remember it, missense, you're missing the right amino acid. It doesn't exactly mean that, just to remember. Missense, you're missing the right amino acid. You changed one of the bases, so you changed the amino acid. Nonsense means you change this, okay, and the new one will mean stop. It's wag, waga. It will be one of those, right? It's changed, it's be UAA, whatever it is. We call that nonsense. To remember it, this is how I remember it. It's nonsense to stop all of a sudden. It's nonsense to stop. It's nonsense to stop. Okay? Silent mutation, that's not bad. Remember the redundancy that we talked about? So you have more than one code. So if it just happened that you switched one code with another code and you are lucky that it means the same amino acid. Can it happen? Yes, it's silent. Silent means basically no bad result. Okay? You're fortunate enough that you changed the code to the same amino acid. Silent. Bad mutation is to mutate the opposite way. You did something was wrong and you actually mutate to the right one. Bad mutation. Frame shift is whether you are deleting or adding, you are changing the whole frame. And what we mean by frame is like this: 1, 2, 3, 1, 2, 3, 1, 2, 3. Right? If I add something here, this is the frame, right? Frame number 1, frame number 2, frame number 3, right? What if I added this here? The whole frame will be shifted. Look, this will be 1, and this will be 1, and this will be 1. Isn't this shifting the frame?
Yes.
And the same if you delete. If I remove this. So here's what's going to happen. This is going to be 1, and this is going to be 1. Did you get the idea?
Yes.
So how can we get the frame shift? If you delete or if you add. In both cases, it's going to shift. Shift to the right or shift to the left, it's a frame shift anyway. Okay? Is that clear? So this is the frame shift. So when you get the frame shift, in which type? In deletion or addition? Deletion or addition. In both cases, you get the frame will be shifted. Like it was A, B, C, D, E, F, G, H, I, for example. Okay? And you remove the C. So it will become, instead of ABC, it will become ABD, right? And now you're shifting the whole thing. Everything after that will be different. And when you make it different, most of the time it's harmful. Okay? Sometimes it's not harmful. Most of the time it is harmful. So the non-mutated, we call it the wild type. Okay? So this is just to practice what we talked about. The big bad cat ate the fat red bug. Okay? Just an example. So this is 3, 3, 3, 3. This is the normal frame. Okay? This is the wild type, the normal frame. So here is scenario number 1. You are going to substitute. Instead of the big bad, this is substitution. It's not deletion or addition, right? It's just substitute. So instead of B, you change this one. Okay? So this will become mad. Bad means an amino acid. Mad means another amino acid. Okay? So this is called missense. You're missing the right amino acid. Just to remember it. Nonsense. The big bad was a cat. A big bad cat. Instead of cat, you change the code and now it means stop. Okay? So that will be the nonsense. And this will lead to premature termination of the protein, and this type is usually the severe type. Okay? The other type, the missense type that we just talked about, it depends. Like you change this amino acid to another amino acid. How bad it is, I don't know. It depends on which amino acid and is it going to change much or not. But this is always severe. Most of the time it's severe. Why? Because you stopped. You're making a bad protein anyway. Okay? Did you get the difference? The missense is not as bad. Frame shift can become from insertion or deletion. And here it is, you insert, delete, and you're going to change the whole frame. And everything from that point, so you started here, right? Anything before that, it was fine, right? Once you added or deleted, everything after that, all of it will be translated into something that's different, right? Which is usually non-functional. Missense, is it severe or not? It depends on, is it bad or not? Is it severe or not? I don't know. It depends on what amino acid did you substitute, did you switch. Termination is bad, always, okay? Which is the nonsense. Frame shift, it's non-functional. You'll make a protein that's non-functional. Can you repair the mutation? Most of the time, you can repair it. You have some enzymes that will try as much as possible to maintain the integrity of these codes. So what else? What exactly do we have that try to maintain? And you have to fail all of this in order to get the mutation, right? So actually, practically speaking, you have a lot to keep your codes correct. You have DNA polymerase, for example. It's going to proofread the nucleotides. So not only that you put the nucleotides together, the DNA polymerase will say, okay, to avoid mutation, let me proofread it. Let me make sure that you actually did the right thing before we move forward, right? The DNA polymerase. You can also repair in a mismatch. If you're mismatching, like, isn't it supposed to be ATCG, right? What if you try to put the A with C? No, this is a mismatch. I'm going to correct it and repair it, okay? So this is if you skipped, like polymerase, I did the proofreading, you're good. Okay, what if you did the proofreading but you missed something and you did a mismatch? I'm going to repair it, right? If you are exposed to ultraviolet radiation, I can do the repair too. So not because you're exposed to ultraviolet radiation, you have to get a cancer. You get a cancer if you did not repair. So you can repair still. Excision repair. If this part is bad, you can excise. Excise means to cut, right? This part is bad, I can do excise. If you failed all of this, try to proofread it. You skipped it. You see a mismatch, repair it. Ultraviolet radiation changed something, and you repaired it. You excise if you skipped all of this, you end up having the mutation. So it will be permanent then. Specifically for the chemical mutagens, there is a test that's called the Amy's test, and this test is to detect the chemicals that have potentially carcinogenic. Okay, so if you're working in a factory or working on something specifically in a factory, they have to do the Ames test. They have to know, do we have one of these chemicals that are carcinogenic or not? If we have it, we need to do something in this factory. If you don't have it, the Ames test will be negative and you're fine. You don't have any one of these chemicals. Usually the mutations will have a bad effect, negative, but not all the time. Sometimes the mutation can be positive. It can make something that's good. Most of the time it is either non-functional or harmful, right? Non-functional or harmful. But sometimes it's not any of those. Sometimes it's actually good. Good for the organism. Like for bacteria, for example. We used to, do you remember that 50 years ago they used to use some antibiotics and they don't use it anymore now? Yes, it's very old. What happened here is that the bacteria, for some reason, got a mutation. And this mutation resulted in the bacteria become resistant to the antibiotics. So the ones that do not have this mutation will die. The one that's got the mutation and become resistant will survive. And these are the ones that are going to stay and continue with us, right? So is that good or bad for the bacteria, this type of mutation? It's good for them, isn't it? It's good for them. So this is a positive one, okay? Let's say fish, for example, okay? We know that in ancient time, there was a lot more ice than what we have now, right? So some of these fish that used to live there, right? When it started to melt down, this is not their normal habitat. They are going to die. But some of these fish, something happened to it, and they got mutation. I'm just giving you examples. This mutation resulted in that their body can take care of that. It changes the temperature. They are going to survive. Isn't that something good for them? So it can be positive, it can be negative. If it results in them to accommodate their environment, this is something good. Good for them. Can you tell me which one of those? D, deletion. What else besides deletion? Insertion. Deletion and insertion lead to frame shift. None of those. Right? What silent mean?
It changed, but it doesn't change. Like it changed the base, but it doesn't change the what it codes for, like the amino acids.
Yes. It leads to the same amino acid. Silent. Missense, you're missing the right amino acids, right? So you change the amino acid. And this is usually substitution. You didn't delete, you switched. Okay? Nonsense, stop. It's nonsense to stop like this. Deletion, if there is E, we say insertion, it will be D and E, right? Just in case. DNA recombination means, I talked about it a little bit, you recombine. Mean you take a piece from here and you recombine it with another DNA. So genetic recombination, and we talked about it before, it can happen automatically, it can happen in the lab, you can do it either way. And this genetic recombination and also mutation, both of those can lead to variation in the microorganisms that used to be identical. What does that mean? Here is what it means. This is a type of bacteria, okay? E. coli, for example. All of the E. coli are the same, right? And then either a mutation happened or a recombination. Recombination is not a mutation. Recombination is you get this patch and you add it, recombine it, right? So whether we're talking about recombination happened or mutation happened, in both cases, you instead of having one type of E. coli, now you have two types, right? It can lead to 3, 4, 5 different types. And this is where we start saying type 1, type 2. Why is it type 1, type 2? In the ancient time, it was only one type, right? Why is it 2, 3, 4, 5 types now? Because either recombination or mutation. Clear? There is an enzyme that's called restriction enzyme. And this is the enzyme that can, if you talk about recombination, like you're telling me, I will take this piece and combine it with another DNA. How did you cut this piece? Restriction enzyme. It's going to cut that piece and the ends will be sticky, so it was going to stick with the other DNA. Okay? In order to do recombination, we're talking about recombination now, it's either conjugation, transformation, and transduction. You have 3 different types. Conjugation, in order to do that, we will talk about it, you need a plasmid and a bacterial sex pilus. A sex pilus is like a sex organ that's going to transmit from one to one. Transformation, this is something that we do in the lab. Transduction, you're using a virus to transduce it. So conjugation, if you look at this picture right there, it needs a plasmid, and you need this sex pilus. And through this sex pilus, a piece from this is going to leave one and go to the other, like this. Okay? You're cutting this piece, go to the other one. It can go from bacteria to bacteria, for example. And what's going to help you to do that is the plasmid. So what do I need to make this conjugation? Conjugation will need 2 things. Number 1, I need to recombine a piece from here, bacteria number 1 to bacteria number 2. I need to recombine this to this. What do I need to do that? If it is conjugation, I need two things. You need a plasmid and you need this 6 pilus. Okay? The 6 pilus is through which this is going to leave, and the plasmid is what's going to transfer to the next one. And then it will go and join the new DNA. So here is conjugation. What is this? This is the 6 pilus, right? So plasmid is here. Plasmid is the one that's going to take this piece, go through the 6 pilus, transfer it to the new bacteria. Okay? Transformation, on the other hand, it's going to be a donor. It's a piece of DNA, for example, a fragment that leaves its spot and go and be transformed into another piece. And this is something that we use in the lab. Here's what you do in the lab. You get that piece from somewhere and you insert it into. From the donor, and you insert it into the recipient. This is usually done in the lab. For recombinant DNA technology in the lab. Transduction, on the other hand, will use bacteriophage. If you wanted to do that, use a bacteriophage. Do you remember the bacteriophage? What's a bacteriophage? It's a virus that infects the bacteria. Yes, a virus infects the bacteria. When this virus infects the bacteria, it will take a piece of DNA, introduce it. When it gets into that, it will introduce it. So you need bacteriophage. You need a bacteriophage. When I need plasmid, not a bacteriophage, when I need a plasmid, what do you call the process? And a 6 pilus. We call it conjugation. So when you're using plasmid, conjugation. If you're using bacteriophage, transduction, transformation in the lab. Okay, a piece in the lab. Can you answer this? Mediated by a plasmid. Using a plasmid is conjugation. Okay, so that's it for this chapter.